All right, let me just put you all here. Start this.
Yeah, that's good.
Can you see it?
Yeah, perfect.
Okay, so I get my clock going here. Okay, so first I want to thank the organizers for putting together this meeting. It's nice to see you, even though we have to do it through a computer screen. Today we're going to talk about metabolic fitness models, and we're going to use them to predict the evolution of antibiotic resistance. So this is work that I've been doing together with Michael Essig at the University of Cologne. And the experiments are done by Omar Varsi and then Anderson at the University of Uppsala. So the work is based on this preprint, which I hope it will materialize very, very soon in its final form. And in a broader context, what I hope I can convince you today is that systems biology can become a very useful tool for evolutionary predictions. I am going to discuss this in the context of antibiotic resistance. So to do that, just a quick recap on that. So antibiotic resistance is essentially a problem of bacterial growth. So bacteria are resistant when they grow in conditions they should not, and that's becoming a problem. So to discuss growth of bacteria under antibiotic challenge, what people usually do is to use these dosage response curves. It's this object that's shown here, schematically illustrated for the wild type. It's starting off at 1 because we are going to use here the growth rate of wild type in absence of drug to scale the growth rates of the study. And here we have two important points that give measures of resistance in this curve. The D50, that tells you how much drug you have to give in order to have the growth rate that the bug had in absence of drug, and the MIC, which tells you how much drug you have to give in order to halt the growth. So what people initially did is that they investigated, they characterized mutants that were selected under high antibiotic challenge. And so under these conditions, resistance was provided by mutations of large effect in target genes. The dosage response curve of a mutant selected in this regime is typically, that's typically selected in this regime is shown here. It comes with an offset in the y-axis, which reflects the fact that in absence of drug, resistance mutations often confer a fitness cost. And then more recently, people focused on the regime of sub-inhibitory concentration. So here, the wild type is still growing. And what they found out is that resistance comes, in a whole lot of different ways. So there is a whole zoo of resistance mutations with different mechanisms, and they confer different levels of resistance and come with different costs. So a summary of these experimental results that helps to understand a little bit of generics of growth is that typically the amount of resistance and the amount of costs are correlated. So this is in the form of a trade-off, but just knowing about this trade-off is not enough if we want to predict evolution, because here, what we would like to know, we need to compute growth rates and resistance levels of different mutants as a function of growth conditions. So the question that we need to address to predict antibiotic resistance evolution is how is it that nutrient levels, drug action, and resistance evolution set growth rates? So to address this question, we did a systematic study of this regime of intermediate drug concentrations. And then we will use the results of our experiments to build on an existing metabolic model of antibiotic susceptibility, which in this picture here of growth rates as a function of phenotypes corresponds to this orange line. And so we build an evolutionary model of antibiotic resistance that is predictive. And what our model does is that it establishes computable fitness landscapes in the drug-free environment, so this line that I'm showing here. So you can predict the trade-off between resistance and cost. So it establishes computable fitness landscapes at a given drug level here. So we can tell which mutations are going to be selected. So this is going to be clarified as we go along. And our model establishes maximum growth trajectories of resistance evolution, here, this line, which lets us predict prevalence resistance mechanisms. So the whole point of the talk today is to fill in this picture and to see how the theory talks with the data. So to do that, the first thing that I'm going to talk about is this framework that establishes a correspondence between bacterial growth and between systems biology and bacterial growth. Then I'm going to talk about the resistance experiments that we did, and then I do predictions. Okay, so bacterial growth. For bacterial growth, we are going to use the work of Matt Scott and Terry Hua, which very recently have established a very simple and fundamental relation that lets us describe bacterial growth in terms of two phenotypes that relate to basic cell metabolism. So here, growth is just the harmonic mean of a nutritional capacity that gives the synthesis of biomolecules from external nutrients, and a translational capacity that gives the rate of protein synthesis by ribosomes. So in this framework, perturbations of growth can be implemented in two ways. So we can vary nutrient quality at a fixed nutritional capacity and check what happens with growth properties. So we would probe growth along these lines, but we can also vary translational capacity at a fixed nutritional capacity. So then we would probe growth properties in this axis. And this is what we're gonna do to use this formalism to address this problem of antibiotic resistance evolution. So the whole idea is that we are going to perturb growth with a ribosome targeting drug. And what ribosome targeting drug do is that they capture the ribosome. So then when they do that, they reduce the translational capacity. So this happens in a drug-dependent manner. So as we increase the concentration of a drug, we expect that growth will move down. So we decrease the growth. So you can just go back to our growth prescription and now describe it, the drug dependence on growth in the same way, but our translational capacity becomes an object that depends on the drug concentration. The only problem is that we don't quite know what is this expression here, this prescription. But this is where the existing metabolic model of antibiotic susceptibility comes in. So very recently, Matt Scott and Greulich and Rosalind Allen, they established a quantitative model to describe growth inhibition by ribosome targeting drugs. And this integrates the mode of action of drug with bacterial physiology. So the model is quite simple. So here, the translational capacity depends on the amount of ribosomes that are free to translate. These in turn will depend on the intracellular drug concentration. So this is intuitive. Then the intracellular concentration of drug is determined by transport through the membrane and the kinetics of binding between the ribosome and the drug, and then by dilution by growth. So when we start talking about these processes, we can then assign biophysical parameters. So now we start modeling the mechanism, and the physiological aspect that is included in this model is that as the drug impairs growth by inactivating the ribosomes, what the cell does is try to compensate it by upregulating the ribosomal content. So the beauty of this model is that everything that I just said can be expressed in terms of two parameters. And these two parameters, they have a biophysical interpretation. So they are a concentration scale and a rate scale. And they relate to transport coefficients and binding and the kinetics of binding between drug and ribosomes. Okay, to test if this model is good for our purposes, the first thing that to do is to measure growth dosage response curve of E. coli subject to streptomycin. So these are the dots here. And we feed the model to the data. And when we do that, we see that the model describes it well. When we have, so in light of this metabolic model now, we can start playing around and trying to understand one or other thing about the system. The first exercise that to do here is to shut down the physiological response to see what would happen. And the unregulated cell would go through this line. So the first thing that we can see is that to a first response in terms of defense mechanism that the cell does is regulation. So we learn that physiological response just by having this biophysical model. We learn that physiological response is a first mechanism based on regulation. So when we go back to our picture, then we fix this point. This is the wild type, and so we have our wild type and how is it that it responds to drug challenge. So now the next steps is to discuss the experiments, and then we see how to complete this picture with these evolution experiments that we did. So our basic experimental setup consists of classical fluctuation assays. So we grow parallel cultures overnight, and then we play them under antibiotic challenge. So here we are going always to use streptomycin. So all experiments are going to be done for streptomycin and E. coli. The point is that if there is a mutant that's resistant to the amount of antibiotic, then it builds a colony that can be picked up for further further study. So one thing that was important for our study was to transform this question from a binary question of yes or no, this bug is resistant to this antibiotic challenge to a more quantitative question. So we performed fluctuation assays and selected for mutants at a range of antibiotic challenge from 0.9 to 7.4 times the D50 of the wild type. And at each of these drug concentrations, we selected 8 mutants that were used for subsequent analysis. Okay, the mutants selected here, up to 5, they had alterations in membrane, in genes relating to membrane functions. And at high antibiotic challenge, what we see is that mutations were targeting a gene, a different gene, so that is related to, that encodes a subunit of the ribosome. And so here we start seeing that there is another different mechanisms that are emerging as the drivers of resistance. So in light of this information, we feed the metabolic model for all membrane mutants, so everything that was selected here, and we extract for each of them the amount of resistance, the fitness cost, and the response parameters that I recall here. They are related and give us information about influx and outflux rates and the biophysics of the drug action. Okay, so the first thing that we did once we had this, once we had these observables, was to check what happens to the observables related to the trade-off. And as we see, there is a very clear relation between the gain in resistance and the cost that you are paying in absence of drug. So what we wanted to do next was to explain this trade-off on the basis of a model. And here, our assumption was that, okay, so we're gonna start going to predictions. So here, our assumption was that as the antibiotic reduces the translational capacity, what evolution is doing for the bug is to adapt the nutritional capacity. The whole idea is that we know that these bugs have membrane mutations. So it could be the case that these membrane mutations were generating a proportional reduction between the drug intake and then as a collateral effect, it would have a cost and fitness in absence of the drug. So here we would relate a phenotype that is depending that we extract from the system under drug challenge with something that is independent of the drug, but they would be related by a single parameter. So if this is true, then this uptake parameter that relates these two phenotypes we should be able to describe how the drug-free growth rate depends on this uptake parameter, and how the resistance depends on this uptake parameter. So these two lines would give our the predictions of our theory, and then when you check it against the data, we see that it has very good agreement. So by the way, I haven't mentioned the different colors here. They are just illustrating mutants that were selected under different drug challenges in that scheme that was presented before. Okay, so we have very good agreement here, like for when we compare the prediction to the data. So now when we plot drug-free growth rate as a function of resistance, we recover our trade-off, and then we have an understanding of what is generated, what's the phenotype behind this trade-off. Okay, so one thing that is very interesting about this trade-off is that it only depends on the evolutionary resistance mechanism and on broad metabolic parameters of the wild type. So it doesn't depend. In order to get this line, we don't need to know specific details of the genetics behind these mutants, just the mechanism. And so we know from here that if a mutant wants to increase resistance by 10%, relying on membrane mutations as a defense mechanism, so we would expect this mutant to pay a 2% cost in drug-free growth rate. So now when we go back to our picture, what the fitness model does is that it establishes this fitness landscape in absence of the drug, but then it also gives us a way of getting all the mutant lines that are these orange lines here. So since now we know how these mutants are growing, so what the fitness model does is that it establishes a fitness landscape at any given drug level. We just need to specify what is the drug we're interested in studying. And the cool thing about it is that it allows us to identify what is the mutant that at any given drug concentration will have the highest growth rate. So for the one that was used here in this line, we have this dot. And now what we get from here in the next step is an optimality principle. In other words, we can now use this framework to identify, this model to identify a specific drug challenge at which a particular mutant with a given uptake rate will have the best growth rate. So this would give rise to our cyan line here and a model prediction. So the model prediction would be a prediction for maximum growth rate as a function of growth challenge. So from the model, this would be the line that I am showing here. So maximum growth rate as a function of drug level. And now, when we compare, now we can use extract the drug level at which each of our mutants would have maximum growth rate. And when we plot it against our prediction line, what we see is again a very good agreement with the data. Okay, so now the idea is that we can start using this to predict optimal mechanisms of the drug resistance if we just compare what are optimal growth trajectories that come from different mechanisms. So the whole modeling is general, so in principle, we can apply it to different defense mechanisms. And so before we started already comparing what was the effect of regulation, and what we saw was that we had a first defense mechanism, but then now the next thing that we do is to compare the growth rate of wild type with the optimal growth rate generated by membrane mutants. And then what we see is that there is a point where when the wild type fails to grow, this would be a very good strategy for defense. But then after a while, this growth rate declines. And membrane mutations wouldn't be anymore something that would help the bug to grow. So here we would expect to see a different mechanisms taking over. We would need a different viable mechanism for growth. And when we compare what we get from the optimal growth rates of target mutations, so for the mutants that we had in our experiment, target mutations were very insensitive to the drug. We had a flat line. So we would see that this would be a viable mechanism to be selected to appear. So now the question is, what would we see in evolutionary experiments or in the laboratory experiments? And so we can integrate information of mutation rates to start building expectations of what are growth rates of which mechanism and which mechanism would be seen so in the lab. So this corresponds to these gray boxes here, and the points correspond to the data. So we see that this framework let us predict different rankings of mechanisms that would be selected in the lab as a function of conditions. So here it's always growth rate as a function of the drug level at which selection happens, and we see again a very good agreement between the data and the predictions and the data. So our theory allows us to rank different mechanisms of evolutionary defense as a function of conditions. So now the last point that I want to make here is that the type of questions that we can start asking is what would happen in a different situation where we wouldn't be able to rely on a mechanism that is based on shutting down this membrane. As for example, if we do selection experiments in a poor media. So here we just consider a situation where the growth rate of the wild type it's one, in absence of drug, is 1/5 of the growth rate of the wild type in the experiments that we had previously. And as you see, so this is a line that would put the detection, it would set the limit of detection in the experiment. So we see that the unregulated cell would be super bad here, and then we would have the wild type growing. But the funny thing, which is interesting, is that the whole regime of membrane mutations goes below our detection threshold. So we wouldn't expect to see this type of mutants in the experiment. Instead, what we would expect from the theory would be to see target mutations already appearing at concentrations of drug that were much lower than than where they were appearing before. So when we do selection experiments under these conditions, so low growth, so poor medium, we see that there is another mechanism based on mutations on the stress response system that appears for mutation for selection at low drug concentrations. And then again, when we integrate the information about mutation rates, what we see is that the theory would be again in very good agreement, telling us what are the growth rates and what mechanisms we should expect to see in the experiments. Okay, so just to summarize, I hope I gave, this was an example that illustrates the fact that systems biology is very useful for evolutionary predictions. So the whole point is that systems modeling is useful to describe the nonlinearities that we see in biological data. And the whole power of this approach is that, as we show here, is that to the eyes of a metabolic fitness model, drug action, environmental changes, and resistance evolution, they are all coupled perturbations of the microbial system. And then because they are all coupled, there are certain constraints that must be respected in order for this bug to be alive and then to survive. And therefore you can leverage on that for building your predictions and expectations. Okay, so this in general can be used for other drugs and other mechanisms. And one particular direction that I'm excited about for future research is how to extend this to microbial communities. So with that, I thank you and I take questions.
Okay, so there are actually many questions already in the chat that I'm going to read just because there are only a few minutes left. So Guillaume Martin asked, are the prediction lines parameterized? Are they partly fitted?
So yeah, so we fit the dosage response curves of the wild type, and there, so of all the mutants. But the prediction lines, they don't depend on any parameter. There is one parameter that we need to know, which is the ratio between nutritional and translational capacity of the wild type in absence of drug. So this can be measured. We didn't have these measurements, so we used the data of mutants to fit that parameter. But yeah, you can get it without fitting. So it's without any.
Okay. Michael Manat asked, how do you measure the uptake rate?
Yes, so we did measure the uptake rate to try to check this assumption. So it turns out that the assay that we use is not very sensitive to discriminate between different mutants. But what we see is that in comparison to the wild type, the uptake rate of mutants is reduced.